All right. Thank you. Thank you for that. I would have totally kept going and not have done that. So, um, yes. So we're going to go ahead and start this our series. We're hosting a Tiny Earth webinar series. We're going to show you how to perform metagenomic analysis of your tiny Earth isolate within the BV-BRC. Maybe you're not part of the Tiny Earth community and you still have metagenomic data. No worries. This is a great spot to be. You will learn how to do clear, direct tasks within the BV-BRC and hopefully be helpful to address the questions for your own particular data. So it's going to be a two-part event starting today, and then we'll meet again next Friday, both at the same time. Quickly, just want to give you what is and what is not in regards to what you should be expecting from this. This workshop is designed for the Tiny Earth partner instructor community. We're using the BV-BRC as an educational resource has been taken into account, meaning um, if you're teaching a Tiny Earth course, the analyses you might be interested in are something that you are probably going to have maybe a small set of students performing, or maybe a larger class of 20 to 30 students. So you might want to learn how to do particular things that's important for the bioinformatic workflow, but you might not need to know about every single thing that you can do within the BV-BRC. That's what it's kind of designed for. Now, if you're not a techie, no worries. You can stick around. I didn't put this first as a mean to indirectly tell you to leave, right? I'm not that friend who's hosting a party and now I'm yawning and looking at my watch, but I'm doing everything but telling you to go home. That's not the case here. Please stick around. Might be different data, but the questions might be the same. So you can definitely learn a lot and take away a few things from this experience. All right. So this is designed to introduce our registrants to multiple BV-BRC services and tools for you to achieve clear, simple goals within your bioinformatic workflow. I don't want to say it's a speed run workshop, but it kind of is a speed run in the sense that you will take away a lot from the different services you'll use, different outputs that are relevant. We won't cover every output within the services that we are going over. We won't cover every service that we offer. But in regards to the questions we're going to address, I think you will know how to do it and feel confident to do it within the BV-BRC. That's the goal that we have here. So workshop goal, we want to learn how to perform an easy yet robust metagenomic analysis within the BV-BRC. I want to know how to do quality control, assembly, annotation. I know that I can use the BV-BRC and I'm confident that I can do it on my own after this experience. So directly related to the last slide, this workshop is not an in-depth comprehensive workshop covering BV-BRC tools and services. Within the hour and a half that we have, I could also spend the same amount of time covering 2 to 3 services as compared to, I think, like the 5 or 6 that we might get through today. Now, if you are looking for a more comprehensive training experience, then I would highly suggest you register for an upcoming free BV-BRC workshop we're holding at Argonne National Lab that is comprehensive. We'll go over split up between bacterial and viral analysis from the first 2.5 days and the last 2.5 days. So you'll get a full comprehensive walkthrough of our services and tools, each bacterial analysis tool, and as well as for viral data. So if that's something you're interested in, please check out the link. I will post it in the chat in a second once I'm done with my slides. All right. If you have questions throughout this workshop, there are going to be brief moments during the workshop where you can throw out questions. You can just turn your mic on if it's quiet. When I say brief moments, when we're walking through these services, I'll give you about 2 minutes to be able to submit your job, upload the data, so on and so forth. In that time period, if you have a question, feel free to shout out and ask. I also highly encourage if something comes into your mind, I'm yapping, there's not a moment for you to ask the question through your mic, please use the chat. We are very lucky to have a group of experts, being the experts means my colleagues, all very much more knowledgeable than myself, who are joining us for this event. Thank you again for being here. using your time to help us out. So we have Rachel Paretsky, University Illinois at Chicago. We have Anna Capria from the JCVI, Rebecca Wadham coming from the University of Virginia, and Nicole Bowers at Argonne, who are all happy to help address your questions in the chat. So, um, all right. So to get started, maybe you're not familiar with what the BV-BRC is. We'll do a quick walkthrough of what we are. So the BV-BRC is the Bacterial and Viral Bioinformatics Resource Center. It's an NIH NIAID funded initiative. Our main goal is to support biomedical research on infectious disease, the exploration, the discovery of pathogens. It's also integrating publicly available genomic data, metadata into our site and providing users with robust bioinformatic analytical tools to use with those data sets. So this initiative has been going on for over 2 decades. At one point in time, it was three separate BRCs, Patrick, IRD, and Viper. And then in 2019, we merged into one unified resource where now you can use our website to perform a collection of analyses, regardless if it's bacterial or viral data you're interested in using. So our mission, as already stated, we integrate publicly available omics data, curate metadata, as well as provide analytical resources for you to use with these data sets that can help support the ever-growing amount of public data and the need for necessary resources. In addition to that, to keep our users up to date with the enhanced technology, we continuously are developing and innovating new bioinformatic and machine learning tools and resources that you can use for discovery of pathogens, drug development, vaccines, and therapeutics. Really, whatever the infectious disease research community might be interested in looking into at any given time. And lastly, but as important as all the rest, we pride ourselves in providing training experiences and outreach to not only researchers, graduate students interested in using our resources for their own personal research, but individuals from government organizations as well as educators, showing that the BV-BRC can be also used within the classroom just as efficiently. All right. Now, if you might not have heard of the BV-BRC, promise, I'm not lying. We have a lot of people that use our resources. We have over 60,000 registered users, 50,000 unique visitors each month, submitting thousands of individual jobs, and we have over 23,000 citations. Our user base is fairly broad, from basic science, applied science researchers in academia, government organizations, nonprofits, pharmaceutical companies. We also have clinical researchers that use our resources, epidemiologists, bioinformaticians and statisticians, data scientists, as well as educators. Several institutions where we have classes that use our resources to teach bioinformatics. All right. Now, as mentioned already, the BRC is constantly integrating publicly available data sets. We have millions of genomic data from bacterial and viral species. We also have direct access to a number of public databases, GenBank, SRA. If you are using our services to upload FASTQs files, you have the ability to provide SRA ascension numbers instead of actually uploading the file itself. It connects to the cloud database. Our service connects to the cloud database using that SRA number, and we can access data that way as well. So data is continuously curated, but you also can store your own private data within our website using the user workspace, which I'll go over in a second. And we integrate all these different data sets with our bioinformatic tools. So our website is bv-brc.org. You can use the QR code you can type into your website. We're for sure going to be using it because we got it. You're going to use it for your exercises. So, but that is our website. That is our homepage. This is our assortment of provided tools and services. We are going to cover a small portion, maybe a third of the actual resources that we offer on our website. So, like I said, this is to get you confident to thinking about using the BV-BRC as a tool that you have access to, that you know you can do particular analyses with, but maybe something else sounds neat that you want to look into further. But now you got your foot through the door, right? So the wheels are turning to help you kind of gain more experience with using our website. Um, all right. And like I mentioned, we have our BV-BRC workspaces. Workspace is essentially like a directory where you can store your own private data. You can make data public and share it with the broad community. You can share it with particular users. You can upload stored genomic sequences. You can store FASTQ, FASTA files. Um, so it's very helpful in regards to being able to collect and maintain your data within our site. Additionally, we are working on developing a BV-BRC, a dedicated BV-BRC educators workspace where you can designate a workspace, make it public, designate it as an educational resource, maybe an exercise of some sort. You can upload your FASTQ files, your sample data that you need to conduct the exercise, upload the exercise material, and then not only can you make it available for your students to use directly, copy it over to their home workspace, but also other educators from different institutions can look through your exercises and determine, this looks pretty neat. I want to add this to my course as well. So they can copy everything over and immediately start implementing it. And this hyperlink here is a view of our educators documentation page. In the near future, beginning of the summer, we'll host another webinar on how to put together an educator's workspace. If you click on the documentation page, it will guide you through suggestions for what you should include if you want to publicize an assignment. All right. So for how is the BV-BRC relevant to your Tiny Earth course? We have a few Teppies here joining us. If you're not familiar with the Tiny Earth, it is a Cure course-based undergraduate research experience that was created by Joe Handelsman, is led out of University of Wisconsin-Madison. The course aims to not only give undergraduate students hands-on experience conducting research within a classroom setting, but also allow them to address a very important and real problem. And doing so in a fashion that allows them to be able to actually provide and discover things on their own, something that is unknown and gain an ownership over these discoveries. Overall, these students will collect soil samples from nearby environments. In their lab, they will culture these organisms and then pick isolates out of them to do a survey of to identify which ones can produce antibiotics. Oh, yes. The essence of the course, the problem they're addressing is the discovery of new antibiotics. Right? I forgot to mention that part. They do this by collecting soil samples. They culture isolates from these soil samples. They create lawn plates where they put their isolates on a lawn. There's a lawn of a gram-positive or gram-negative bacteria. They add their isolate to it. If rings of inhibition form around their isolate, this tells them that this isolate produces an antibiotic. And then they go on trying to identify what that isolate could be through a number of mechanisms, including lab biochemical tests, 16S amplicon sequencing, PCR, as well as at UIC, we use a biolog metabolic profiling assay for them. Now, most courses, I think, depends on the expertise of the instructor and how they choose to design their course, but they most likely follow a similar theme of collecting soil samples, finding isolates, and then characterizing them in some way. If you were interested in using the BV-BRC, that means you are interested in adding some bio or some genomic bioinformatics component to your course. And that could fit in well within Module 3 when you're characterizing your isolates. Um, you could, depending on the size of the course, how much time you have, you could perform metagenomic sequencing, be able to look at your isolates a little bit further, as well as get a clear image, a clear identification of what your isolate might be. All right, so as I just mentioned, where the BV-BRC can be integrated into fit well within Module 3 of your Tiny Earth research flow. Like I mentioned, if maybe your course, you have a certain amount of time in the dish, adding a bioinformatic component to your course, you would need weeks that might not be there. Totally understandable. Maybe you might have a few students afterwards that are really interested in doing something further with these isolates that they were able to identify and collect. You could have an independent study or advanced tiny earth course where you then go a little bit deeper into the genomics or the bioinformatics component, having students address particular questions related to uh, using that genomic data, as well as ongoing Tiny Earth and genomics courses I've seen that are posted within the TEPI website, tend to be more uh, computationally intensive. They usually go over more technical workflows using Python and R, but maybe you'd want to focus your workflow on particular things, doing assembly annotations, but maybe more complex tasks that are not as easy to perform in your CLI. You might want to use the BV-BRC for building phylogenetic trees, comparative genomics, so on and so forth. So regardless of where if you want to integrate it into your course, into something secondary, or into your genomics course, there's a way you can fit the BV-BRC. And why use the BV-BRC? One, it's free. You don't have to pay for nothing. Sign up. You have access to all of our resources, our data. Students have access as to it as well. It's accessible. It's easy to use. It's a little learning curve, but this is the whole point of what we're here for, right? We're going to give you the basics to how you get started, and you should be able to hand our handout to a student, and they should be able to perform these different bioinformatic analyses. You don't need any technical experience to use our resources. Everything is maintained through Argon's high performance cluster. So you don't have to worry about allocating resources, thinking about computing nodes, how to provide these to your students. We take care of all of that. All right. And that is our background. We're going to now continue on with the exercise itself. The overall goal for today is going to be getting our FASTQ files. We want to make uh, assemble them into contigs. Well, I'll start off. We'll just follow through with how I've been organized here. So we will obtain our FASTQ files. We'll perform quality control using FASTQ utilities. I also mentioned in the handout how you can get some of these outputs within your taxonomic classification service. We're going to characterize our microbiome since we're using we have a metagenomic data set. We use the taxonomic classification service. We can look at alpha and beta diversity metrics, look at the abundance on ID, species, and gender within our data. And we can also, if needed, be able to produce new FASTQ files containing either the classified or all of our mapped reads, or maybe you want to make a file of all your unclassified or unmapped reads that you can use in BLAST, so on, or something along those lines afterwards. We'll cover those things. We're going to construct metagenome assembled genomes, MAGs. We're going to use the genome assembly service to assemble contigs from our FASTQ files. We'll then use that FASTA file of contigs, upload into the metagenomic binning service, which will then cluster our contigs into genome bins and create our metagenomic assembled genome. All right. Um, so, and lastly, we'll cover something that's going to be a little extra. It's not per se related, but still something you might be interested in. Maybe you're interested in looking at antibiotic resistance genes within your metagenomic sample. We can use the metagenomic read mapping service, which will align our reads against or your contigs against the comprehensive antibiotic resistance database, and that will be able to tell you which antibiotic resistance genes have been identified within your data set, as well as the abundance, I guess you would say, the coverage of each gene as well, to give you idea of which ones might be more prevalent within a particular sample. We also have a predictive model that within our genome annotation service that can provide predictions for antibiotic resistance or susceptibility of your isolate. With this, it is in the handout. We'll go over it when we get there. There's a list of genera that are included within this model. If your organism is one of those genera, if you run genome annotation, you'll get the output, the predictive model output that'll tell you antibiotic resistance and susceptibility for that organism. But it won't be important. It won't be necessary for our exercise, but I'll include it just in case, something you're interested in looking in further later on. All right, very cool. Now, excuse me, at this point in time, you're going to need to do two things. I forgot to mention earlier, if you haven't registered for the BV-BRC, good time to do it now. You'll need it to access the public workspace containing the exercise data that you'll need. So you can open up your web browser, open up the bv-brc.org. We'll go over the homepage in a second. Two, in the email that I sent this morning, contained a handout for our exercise for day one. In depth, we're going to walk through this handout together. So you can open that up as well, and then we'll get started. Now, a couple things. I mentioned the BV-BRC educators documentation page. Just wanted to briefly show you. You can access it with through the hyperlink, but it goes over introducing the goals of the BV-BRC for educators, how to create and publish an education workspace, recommended material to include, documentation, job outputs, so on and so forth, how to let us know that this education workspace is available and up for you to share. You can email us using the contact drop button on the homepage. And then we can publicize it within our list in our documentation page to share with others to know that it's available for them to use and to check out. All right. Now, little background on our isolate. Right. So the data we're using, actually very high quality. We're going to make some great mags with our data set. Now, this data, everything was completely put together by students. I got permission by Andrea Bassett at UIC, shout out, to allow us to use an isolate, to allow us to sequence an isolate to put together this material for this workshop. The strain was isolated in the spring 2025 semester at UIC's microbiology lab course. Students performed gram staining of their isolate. We know that it's a gram-positive rod-shaped bacterium. Within the handout, our exercise starts on page 4. Within the handout, I include all the results for the biochemical tests. You can look at the results for the biolog metabolic profiling assay. These colonies were stored in glycerol stocks, froze. We were able to access those. And we use Plasmidsaurus to sequence our isolates. We sent them pellets, and in return, they did perform whole genome sequencing, sent us nanopore reads, as well as contigs. They do assemble. So in case you're interested in looking in further in the service itself. After that, you can look at the biochemical data. We're not going to go over the data itself, but maybe if you're thinking about your course, thinking of ways that you can use genomic data to validate some of your biochemical tests that you do in the lab. You can use the data I provided, as well as you'll have permanent access to the sequencing data, and you can do a little bit of exploration, maybe to see if If you are isolated as a lactose fermenter, what genes can I find within the BV-BRC that I can see with using some of our services. All right. Now, to start off, as I mentioned, let me see. I'll actually show you the home page. All right. So we're on our home page. Overall, we have uh, drop-down menus that you can use to either look through and broadly search organisms, publicly available data sets for bacterial and viral pathogens that we have stored within our system. You can do searches of different taxa, genomes. Maybe you want to find a particular cake pathway or subsystem within our website. You can do a search before looking for that cake pathway within your organism. Our tools and services is where you're going to access all these services that we'll be using for this workshop. Workspaces, as I mentioned, are your private and public directories where you can store your own data. We also have a public workspaces uh, hub where you can access all the currently publicized workspaces as well. Share things with your students, with your colleagues. Under help, you can access our documentation page. This contains all of our quick reference guides, tutorials, video links to our YouTube where you can access videos for how to use all of our services, um, including information on upcoming webinars, things along those lines. Now, under About, you can learn a little bit more about the BV-BRC. If you're an educator looking to publish some data or publish a workspace, an educator workspace, you can click on Contact Us. Let us know that you're interested in publishing an educator workspace. Send us an email, the name of the workspace, and we can go ahead and put that on our list within our documentation page. All right.
Jamal, we have a
Yes.
question. Could you show them again how to access the education page? That's the question in the chat.
Okay, yes, the documentation page. So we're on our homepage for the BV-BRC. You can click on documentation website. Um, in here, you can see our sidebar of help resources. You can click on education, and that will bring up our BV-BRC for educators documentation page. Has all the information that I had mentioned earlier in regards to making your educator's workspace. After this, I'll also share with the recording the slides that we covered today. I'll also put the slides in the public workspace for the webinar, so you'll have access to those hyperlinks if you want to click on them and view them later on. Right. Cool. Thank you, Rebecca, for letting me know. All right. So at this point in time, you have registered for the BV-BRC. Now you want to have access to our exercise material we need for today. You can do that within your handout. We're looking on page 8. We see getting started, registering for the BV-BRC, accessing the exercise material. After you've registered and signed into your account. Number 2, there's a hyperlink that says you can access the exercise material here. If you click on the hyperlink, it'll open up a tab that has 3 subfolders. We're now in the public Tiny Earth webinar series, public workspace. Now, to make it easier for yourself with accessing the material, you can click and highlight these 3 folders. What we're doing here is copying this over to your own private workspace. All right. In this green action bar, you can click on copy. You can store it wherever you would like. I like organization, so I would recommend creating a workspace, title it whatever, on your workshop. Cool. All right, create a workspace. You should see it pop up on your screen, open up the workspace, and then you can click OK to copy those 3 folders over. On the bottom corner, it will tell you when it's finished being copied. And if you were to go back to your home page, I did that by clicking on my username. Oh, well, technically it's going to have my username. So for you, you'd want to go to workspaces in the drop-down menu and then click on home, or click on, sorry, my apologies, click on my workspaces. And in that list, you should see the workspace that you just created, and it has the three folders that are copied over. Uh, Now you have everything you need to go ahead and get started with our workshop for part one. And then I'll have a brief moment after we submit our FastQ utilities job. For part one, like we mentioned, we're starting off with quality control. We won't be performing trimming for our samples, our sample data set, but might be something that you're interested in yourself. So you will, from the tools and services under utilities, you could see FastQ utilities in the drop down. You can click on the hyperlink that'll open up your FastQ utility service. Now, within the parameters box, here is where you actually are going to be selecting your output folder where you would like your output to be stored. I like it, like I said, organization. I like everything to be together. So I go to my workspaces. Um, hmm. Well, let's see. Should pop up a list of your workspaces. You can access the one you just created, or you can store it here in the meantime, move it over after after your job's finished running, and you can find it in your list of workspaces. Um, and you can select that as your output folder. You want to give it an appropriate name. Usually we'll include my sample name. I'll include something about what service I'm using, and you'll have that there. All right. Under pipeline, this is where you will be selecting which actions you would like to perform with this service. Here, we're just going to be using FastQ or FastQC for our quality control. You select FastQC under pipeline, select the drop-down menu, click the plus sign, and that will add it to your uh, pipeline list. All right. But maybe you're interested in, I don't know, with your own data, trimming, which will look for adapter sequences at the beginning of your reads to remove, paired filter, which will filter out if you're uploading paired read files, it will filter out any reads that don't have a counterpart within the respective opposite file, right? So any forward reads that don't have reverse, any reverse that don't have forward. Or you can perform alignment of a target genome, and you can also remove human genomic data from your reads. All right. For read file, you want to click on your folder next to read file one. We're using paired reads ourselves. We go under my workspaces. You'll find that folder that you just created with your sample data. Oh, and we have raw reads. My apologies. We're going to do, I said paired read. We're actually using single read library. So under single read library, under platform, it's currently selected as Illumina. You want to click Nanopore, and same thing. Click on the folder, My Workspaces, find that folder you just created. Under sample data, you want to click Raw Reads FastQ. After you're done, you want to click the arrow in the top right corner. That adds your library, your FastQ file uh, to our selected libraries. But if you weren't doing single read, you could also do pair read. And as I mentioned during the slides, you can, if you have already uploaded your data or you're using publicly available data that's stored within the SRA uh, database, you can get their SRA ascension number uploaded here, click the arrow, and it'll add it over as well. Once you're done, you want to hit submit. All right. And at this point in time, I'll give you another moment. I'll say minute 2 minutes. It's 1:44, 1:45, almost 1:46. We'll get back started again. Should give you some time to find what you need to find. If you have any questions, feel free to shout it out. If there's anything in the chat we want to bring up, it's a good time to. 44.
Hey, Jamal,
Yeah.
someone wants you to show them how they can get to the FASTQ page again. Can you show them
Yes.
from the top?
Most definitely. All right. So we are on our homepage. We want to use FastQ utilities. We go under tools and services, which will have all the services we're going to be using for our the today and next Friday's workshop. Under utilities, you'll see FastQ utilities. You can click on that'll open up your next window. Um, and this is
And
how we get to our
can you also step through migrating the data so they can access that from this page for the sample file? They're asking
oh,
about that.
like how to copy it to
Yeah, how to get it over.
okay. Yes. So there are, I think the easiest way I would show if at this point in time you would be looking at your handout, your exercise handout for today. We're in part, I think I said page 8 under getting started. Number 2, where it sees you once you registered, yada, yada, yada. Exercise material can be accessed here, hyperlink. That's where we have the public workspace files um, for this workshop. Should open up a tab. You should see J Sharif. It's my username, and then you should see Tiny Earth webinar series in the top as your path to this workspace. There's 3 folders. I think you can figure out. I use a Mac, so I use Command to highlight multiple folders at once. It might be Control or something. If you don't use a Mac, but you can highlight all 3 at once. Or highlight all 3, you can do them all at once or individually uh, later on, if that's what you prefer. Click on copy. Now I go my workspaces. I like organization. You want to create a new workspace, title it Tiny Earth Workshop, whatever you want to title it, to where you can store everything for this day and day 2. I created TE Workshop. Double click it and you would hit OK, and that will copy over that material over to your private workspace. Now, to access that, we do that because it contains our FASTQ files, our FASTA files that we're going to be using for these 2 sessions. All right, you're using a single read library for FASTQ utilities. So you can click on, we're using nanopore sequences. So you can click on nanopore from the platform. Oh, I'm sorry. You want to click the folder for read file. Find your folder that you created. Under sample data is where all the data you'll need, your FASTQ and FASTA files will be. And you'll click FASTQ, and that's how you upload it. So no problem. All right. Okay. So is that Manuela? Were you able to copy that data over? Were you able to access it? If you click on the link and it's empty, then you might have to sign in to your account.
She says she lost you at the very last step.
At the very last step, as in
So I think
for,
when you were migrating,
oh,
from
okay.
start at Nanopore and just, I think it's
Okay.
navigating.
Okay. Yes. So the very last step, then we are, okay. Yes. So the very last step, we are uploading our read library. So you would select Nanopore, and anytime you want to upload data to a service, you'll click on that folder. Here I'm already in the folder, but say I'm in my workspaces. You're looking for the folder that you created where you copied over that material. Under sample data, you'll find all the data that you'll need for the workspace. After you upload it, you can hit on this arrow in the top right corner. In that corner, that's what adds it to your selected libraries. So once you have the pipeline selected, output folder, output name. Okay, wonderful. Then you selected libraries, you hit submit, and then now we'll submit your job. Uh, Now, at this point, ideally you would be just did this analysis on your own, submit the service on your own. You would want to access your results. Here, if you click what I just did from the page I was on, in the bottom right corner, you see the jobs like tab. You can click on the jobs tab. You can also, under my workspace tab, the workspace drop down menu, under data, you can click on my jobs. It'll bring you to the same page, but this will be the list of jobs that you've submitted. Uh, um, so this will be the list of jobs that you've submitted. It'll tell you if it's running, if it's in queue, and once it's completed, you can double click it to access your job results. If you need to resubmit it, you can click on rerun. You can report an issue if you're having troubles getting a service to run successfully. But in this case, so to help us save time, what we are going to do is you copy those three folders over, right? So in your new workspace, you will go to job outputs and have all the job
Jamal,
outputs. Yes.
we have a couple people who are having trouble submitting. If you go to the
Like,
page,
mhm.
so to submit a job, you're going to have an output folder selected and an output name along with the data moved over to the selected library and the pipeline selected. So all of those will have to be set for a job to be able to be submitted. So I would urge everyone that's having issues to make sure that you have those parameters set. And if you
Yes.
still have those parameters set and you are having issues, let us know.
Yes. Very, very important. Thank you so much for adding that. Um, So just to one more time go over, we want to select your output folder where you want to store your results, likely in the folder you just created. Output name, something specific so you know what it is. Make sure you have FastQC selected in your pipeline. Hit plus to add it. We're using single read library. You want to select Nanopore, select your read file, click the arrow to add it to selected libraries. And then submit should pop up blue for you to click on, then it should work well. Right on. All right. So at this point in time, we want to go over what to do once you have your results. Now, save us time, so you don't have to wait. We'll have results already stored within the exercise material. So you go into the folder you created, click on job outputs. Number one, FASTQ utilities. You can click on your job. Results are always going to be designated by this flag emblem next to them. Double click on your results. Um, all right, so we double click on our results, and then you will either you can click on the HTML file, click on view in this top right corner here, or you can click on view within the green action bar. Either way, it will open up your FASTQ report. All right, so now you just conducted a quality control over your FASTQ file. Provide you basic statistics, including the type of file, the number of base pairs, the number of sequences, the length of your reads, and then you can access your graph that will show you the average quality score per position within your read files. General, you're looking for above. Here we have this green area being at 28 and above. So usually 28-30 is where you would like your average quality score to be. If it's less than that, then you'll have to ask some questions. Sometimes it might be at the beginning. You might need to remove a adapter sequence. Maybe at the end, quality tends to degrade, especially in reverse read sequences. So you might need to do some trimming. But yes, for how to save it, I would right click, go to save image as. And that's our first service. So if there's anything in the chat, about FASTQ utilities, please let me know, shout it out. If not, we're going to move on to part 2. All right, very cool. So part 2 is on page 12 of your handout. Now we're interested in characterizing our metagenomic data set. All right, so to do this, we're going to use the taxonomic classification service. We're going to use this to characterize diversity within our data, within our file, our read file. We're going to also be able to look at not only which taxa are present, but the relative abundance of that taxa within our metagenomic sample. And this could provide information, helpful information, not only on the diversity of the sample itself, but maybe if you're interested in looking or in constructing a metagenomic assembled genome, right, you might want to consider looking at how many reads fall into a particular genus or particular species. And that can help guide you in which one you would like to target to construct these mags. All right. Now, to get to taxonomic classification, tools and services, under metagenomics, click on taxonomic classification. Same thing, we're using a read file here. So, all right. So to add our file, you'll do the same thing. So you can see it's going to be quite repetitive on how to upload your data. It's the same way. It should become the easiest thing about this because you're going to do it the same exact way, as long as it's a read or a contig file. So you'll go to sample data, you're going to upload your raw reads. Here we only have one file, so you can leave the name as is. But this service also accepts the ability to upload multiple FASTQ files, and it will look at each as an individual sample. So if you had more than one, then you would probably want to give it a good name to help you identify what that is later on. Once you have it selected, make sure you click on the arrow to add it to your selected libraries. Under parameters, this service does have the ability does accept amplicon sequencing data, particularly 16S RNA sequences. But here we're going to be using whole genome sequencing. We're going to, under analysis type, keep microbiome analysis. We're going to use the BV-BRC database. We're not going to filter host reads, but you might be interested if you didn't do scrub human FASTQ utilities. You can remove host human reads here. We're going to keep the same confidence interval. And the next thing, we won't be saving any classified sequence files or unclassified sequence files. But this might be something you would be interested in doing on your own, particularly if you were an educator looking to perform certain, bioinformatic exercises with the BV-BRC. Maybe you want to have students, we've also, from personal experience, maybe you want to use 6 metagenomic sequence files for a particular exercise. You want them to use taxonomic classification service. Turns out each one is like 2 gigs. So to help students be able to get the same results, if you click on Save Classified Sequences, assuming you're running this exercise on your own before you introduce it to students, it will save all your mapped reads within a new FASTQ file. You can then use that file as your exercise file. It'll decrease the size of your FASTQ file, and it will also decrease the runtime for students. So sometimes students can see things running for 20 minutes. They're like, what is going on? It's been way too long. It's been an eternity. So help reduce the stress. You can reduce that file size. Won't change any analysis results, won't change which mags you'll be able to construct, just to highlight that. All right, last thing, select your output folder. Same thing as before, select the folder where you want your output to go, give it the proper output name, and I'll give it another minute and a half. So recap, taxonomic classification, input file, single read library, same read file that we did for the previous service. Give it a good name. Click on the arrow to add it to your selected libraries. Parameters, whole genome sequencing. microbiome analysis, BV-BRC database, not filtering host reads here. Confidence interval, keep the same. No, no for saving new files. Click your output folder, output name, hit submit. All right. So if there are no questions, we are going to continue on to looking at our results. So on your own, you know how to find it, find your results to look at later. If you click on the jobs box in the bottom of the screen, but you can return back to that job output folder that you copied over. Number 2, taxonomic classification is where we have our results stored. And this will, you double click that and it will open up the results for your taxonomic classification job. All right. Now within here, just to point out a few things, if you were interested in looking at diversity of your sample, we have two different outputs that you can look at. One in HTML, alphadiversity.html, which you can view within your browser. If you double click, or you can click on the view button within the green action bar, and it'll give you alpha diversity metrics, Shannon diversity, Simpsons reciprocal, Fishers for your sample. If you had multiple samples, it would continue on how we see this list of values here under the sample name. If I had 2 or 3, it would look the same except sample 2 name values, sample 3 name values, and it will also provide you a heat map that will compare beta diversity between these samples. CSV file, same thing, you can view within the browser, but maybe you'd want to copy it over and do something within your own computer. You can download it this way, click on download from the action bar to access it. Now, for looking at the relative abundance of our sample, within that folder, I just clicked on taxonomic classification service, multi-QC report, HTML. Now, I click on it, it will open up a comprehensive report of the service. It will include general statistics. It'll include plots for our Bracken results. The service uses Bracken and Kraken2. Use k-mer-based alignment of your reads against our database to identify taxa that are present within your sample. Now, this is helpful for students if you want to have them put together an easy stacked bar graph within our top taxa, while I'll only show the top 5. Maybe you want to look at genus, family, order, phylum. Maybe you want to, instead of looking at read count, you can also look at relative abundance. And they'll change it from number of reads to percent relative abundance. You can also do that as well for your Kraken data, and it'll include at the bottom more results in regards to your FastQC plot. Now, I mentioned briefly before that earlier on that you would also get quality control results within the taxonomic classification service. If that was something you want to look at, the same QC plot we looked at for FastQ utilities, there should be a folder, a directory of the sample name for your read file. You can double click that folder, and then you'll see FastQC results, which you can double click. You want to go to raw reads, and you can open that same HTML file that includes the FastQC results. As you'll see, there's some services that do multiple things, and there's some services that do particular things. I want to show you first how to do something particular. So you know how to do it. You can set the parameters, understand it a little bit deeper, but you will see that there are going to be times where like this, where you have taxonomic classification, but also quality control reports that are included. Additionally, if you want to access your Bracken or Kraken outputs, you can view your output TXT file or report.txt. You can download these, build on them on your own. If you want to do something a little bit more technical, make different figures, absolutely. Go ahead. All right. Now, the next thing I wanted to show is two other illustrations, visualizations that this service produces. The one that we are going to look at here is our sandkey.html file. I really like this image. It gives you the number of reads for each classification, all the way down to species, if it's able to identify at the species level. So why I want to point this out, right? As we mentioned, maybe you're interested in building, constructing a mag from your metagenomic sample, right? Maybe you don't per se know exactly which one you want to construct. You can come into the Sandkey plot. Here we see out of 197,000 reads that align with bacteria, 95,000 of those line to this particular Arthrobacter species, D5-1. 142,000 fall within the Arthrobacter genus. So this would probably be a great target, right, for the construction of our mags. All right. So we went over, all right. We already went over our alpha diversity results. So at this point, we are done with part 2 for taxonomic classification. If there's any questions in the chat, we can look good. All right, very cool. Also, at the very end, once we're all done, I'll stick around. So until everybody's finished, if you have any questions, I'll be around to ask at the end as well. So, all right, now part 3, we are now moving on to constructing our metagenomic assembled genome. Like I mentioned, there's 2 parts. We're going to do genome assembly to construct our contigs, and then we're going to use metagenomic binding service to construct our metagenomic assembled genome. To do this, same thing, go into Tools and Services drop down. We're going to go to Genome Assembly. As you can see, there's going to be some overlap. For our input data, we're going to do the same thing. We're going to go to Single Read Library. You're going to find that folder that has your sample data in it. You're going to upload your FASTQ file. Once you've done that, you want to click on the arrow to add it to your selected libraries. For parameters, you can keep it on auto. The service has the ability to identify if your reads are long reads and short reads, and will choose an appropriate assembler for the read type that you have. If you're interested in using a particular assembler, you can look at the drop-down list and choose one that you want to use specifically. Under output folder, same thing. You want to find the folder that you created where you would like your output to be stored, give it the appropriate name. And something that is important, if you were working with nanopore sequences, under the advanced drop-down menu, which by the way, we're working on nanopore sequences. So we're going to do this right now. Under the advanced drop-down menu, under read processing, you want to deselect filter long reads. Since we're working with long reads, we want them all to be considered within our assembly. It's the most important thing. You can leave all the other parameters the same at this point. Just make sure you unclick filter long reads before you hit assemble. But at this point, you've already done all your other parameters. Your assemble button should pop up. You can click assembly and wait for your job to finish. I'll give you 1 minute. If there's any questions, anything you need me to show again, to walk through, be happy to do so. All right. So if no, double check. Oh, okay. All right. So we have no questions. Then at this point in time, you submit your job. We're going to go on to looking at our results. You're going to find those job outputs already put together. Now we're at number 3, genome assembly. I'm going to click on your job results folder. Here we're going to have we're going to have one folder, 2 files within our output or results folder. Or sorry, or results directory. But the first one is our name of your read file contigs. And this is our FASTA file. You can download this, use this with another service on another website. For our website, you can store this. You can keep this here. You can move it to your sample data folder, wherever you would like to have it. But this FASTA file is what contains your constructed contigs. All right. We also will have an assembly port that shows a bandage plot of your contigs. Here, yours should, they're the same thing. It's my result. So you should see one very large contig and 3 smaller contigs. Tells you which which assembler it used, how long the assembly took, how large the file was. Tells you filtering parameters, and the number of reads, number of bases, the average length of your config. All right. Now, sure. Yes. So, guess there would be 2 ways. I also show you at the very end how to upload this config file. So say you downloaded this, maybe you have a config file already. You could download this to your own computer. You could also take this file, you can copy it to your folder, put it in sample data. Maybe you have a config file that you would like to upload yourself onto our workspace to use with our services. How you would do that? Now I'm in my sample data folder. I want to upload a new file. I could go to upload to folder. I go to select files, or my apologies. One more thing you want to do before you click on select files. You want to go in your drop down, you want to select contigs. This will let our system know that you absolutely need to upload some form of a FASTA file. Then you can click on select files. It will bring up a menu and you can access find your FASTA file, select it, and then start upload, and that'll upload it to your folder here. So,
Jamal, can I interject?
yes.
So if you close out of there and have your FASTA file selected,
Mhm.
if you didn't select contigs, you are able to change the type there. So if you see where it says type contigs, so if you forgot that or you upload something wrong, or you can change the type later, just so
Okay.
you know. That's something
Yes.
it took me a while to figure out. So I just wanted to share
No,
that with this.
thank you. Thank you for adding that. So just to reiterate with the upload button, she's referring to, say you selected your file, you didn't choose it for it to be a config file before you selected the file. You forgot, you uploaded it anyways. Here it would look like it would say unspecified instead of saying config. So if you want to label as a config later on, you can use that drop-down menu to access it and find contigs. So thank you for adding that. All right. So now we have our contigs files. You know how to save it, how to download it. We're going to move on to our next service, metagenomic binning. Under tools and services, access metagenomic binning service. Here is going to use our assembled contigs to cluster them into genome bins. So start with, you want to make sure you have assembled config selected. Click on your folder. At this point in time, you could also still upload from your own personal computer into a workspace. We already know where our data is, so we can go to sample data within that folder you copied. You can find our contigs.fasta file. Under parameters, you could use, let me see what I say. Under parameters, you want to select bacteria and archaea. Output folder, same thing, done a million times at this point. You want to select the folder where you want your results to be stored. Genome group name, right? Since we're constructing MAGs, what's going to happen after the service is finished running? It's going to create a genome group that contains the bin that you just constructed. That way, it can be used directly with other server comprehensive genome analysis and other services that accept genomes and genome groups. So, yes. So you want to give it the name that you would like to give it. And then afterwards, hit submit. So I'll give you, let me check, see if there's any questions. So there's a raised hand, lower hand. Did I raise my hand?
Yeah, your hand's been up.
Oh, okay, thank you. Right. All right, so it looks like we don't have any questions. Now we want to look at our job results. You know what I'm going to say already. You want to find your job output folder. Number 4 for metagenomic binning. We want to select the metagenomic binning job. All right. Here, the first thing that we want to look at is our binning report.html file. All right, so you can click on the file, want to open up, click on view, and this will give you whatever bins are constructed from your metagenome. It'll give you the quality of these bins. It'll give you a summary to tell you how many of these were good, how many were bad. We have one good bin. It gives you the criteria for what we consider a good bin to be. Here we have 100% completeness, 99 course consistency. We have a pretty good MAG. And this is completely, like I said, this was isolated by students. I didn't do anything before other than collecting pellets and then shipping them off. So they did a really excellent job. All right. Now, if you were to, it tells you the reference genome that I used to construct this bin. If you click on this value here, it'll open up another tab that will tell you the reference, bring up the genome viewer, which we're going to go over next week. But it gives you all the comprehensive data about your reference genomes, the contigs, pathways, subsystems, proteins, features. But you also have one for the genome, the bin that you also just constructed. If you click on the value under genome ID. And this is now my bin, my metagenomics assembled genome viewer for the one we just constructed with the same information, still loading, but we're going to go over this in depth further next week. And then it's also, you'll see next week how it has been added to your genome as a genome group for you to use to upload for our upcoming services. All right. Now you already have a FASTA file, but say you used reads instead of contigs for assembly. Maybe you made multiple bins. This will create FASTA files for each bin that was constructed by this service. Anything that wasn't binned will be added to our unbinned FASTA file. So I'll also show this is this is for our specific bin. We only have one, so our report here, if you were to look at the genome report, I clicked on this flag. I'm in my results folder. I clicked on the flag icon for my bin I just constructed. This will give you all the assembly outputs for that particular bin. We can also click on the genome report. It's very similar to the binning report that we looked at earlier. Now it's just for our respective bin that we're interested in. Gives you a little bit more statistics about the bin, information about particular roles that might be problematic with, which is just, let's say if we looked at genes that we expect to be within our genome. If this deviates in some way from what we expect, it can be included within this problematic roles table. So for example, this first one, we predict 1, but we annotated 2. And it'll tell you where it is, what it's close to, and so you can look at it further on and determine if something you'd like to consider further, if it's going to be an issue, or if it's all right. All right. So as I said, we will go over the genome viewer next week. And these next 2 parts, this is everything that you need for next Friday. We've done quality control. We looked at the diversity of our read file. We know we have good quality reads. We know the abundance of which taxa we have within our file. We then use that. We'll see next week when we do comprehensive genome analysis. We can use the genus that contained the majority of our reads as our target genome to perform our comprehensive genome analysis on. But yes, so we identified relative abundance of taxa, looked at our plots. We then assembled contigs from our FASTQ files, and we built our metagenomic bin. All right, so now part 4, as I mentioned before, shows you a few extra things in regards to if you're interested in looking at antimicrobial resistance genes within your read files. Maybe you want to obtain results for that the antibiotic resistance susceptibility phenotype predictive model. So the first one I'll show you quickly is going to be our metagenomic read mapping. Let me see if I have. Okay, cool. So for metagenomic read mapping, you will click on Tools and Services. Under Metagenomics, click on Metagenomic Read Mapping. For single read library, you want to click the folder, same thing as always. Upload your input file. That's going to be, in this case, our FASTQ file. Single read. We can add it using the arrow for parameters. We're going to use predefined list. Although you could upload your own FASTQ file or use a feature group, a group with that's designated within the BV-BR system you created, or yes, that contains a set of genes that you're interested in aligning against. So for predefined, we're going to be using CARD. We also have virulence factor database. If you were interested in looking at virulence factor genes instead of antibiotic resistance genes, I'll click on your respective output folder, output name, hit submit. Look at our results. Let's see. Went to the job output folder that I have already prepared. If you want to look at your metagenomic read mapping results, there's two outputs that I'm going to point out. The first one is your HTML file. So this contains a list of, gives a template, the card nomenclature that you use to designate that gene, gives the function of the gene or the name of the gene itself. It gives the reference genome that it was taken from, and then it gives you these different statistics about the from the service. The score being, the higher the score, the more prominent it is within your data set, within your sample. The score is going to take into account your p-value, but also it's important is your depth. So the depth is the number of reads that are covering that aligns underneath that particular sequence, that reference sequence. The higher the depth, the more you could say abundant it is within your data set. The other file I'd want to show now, the HTML is a good file to look at if you want to have a little bit more interactive with your table. You can look at the kma.res file. Usually I think says unspecified here. So to make it to something that you can interact with within the web browser, under that type drop-down menu, you want to select TSV, hit save, and that'll let you, if you click on view, open it up as a table within the web browser. Click first row contains column numbers, and then you can if I want to look at score, right? Organize it from highest to lowest. I can reorganize my list of AMR genes that were identified and look at it that way. So, all right. And for the predictive model, we use the genome annotation service. Here it accepts contigs. So you would upload your contig file, annotation recipe. We use bacteria. If you're interested in bacteria, you might want to use something else if you're not. But well, actually, if you're doing this for the predictive model, then you're interested in bacteria. That's the genomes that we have. So in your handout at the top of, let's see, it is on page 25. Table 3 contains a list of species that are included within this predictive model, the data set that was used to create this model. As long as your species aligns with the genus of one of the species that are listed in this table, you can get the output for the predictive model. All right. So, for example, you maybe don't need Pseudomonas aeruginosa, but if you have Pseudomonas something is the organism that you have, you can click use for taxonomy name. You can choose Pseudomonas, click on genus bacteria. My label as for your respective file. So give it whatever, Bill is my sample name. So provide that, click the output folder, click on annotate, submit your job. And let's see if I have, oh wait. So for this one, because I'm not working with an isolate, working with a species or genera within the database, I wouldn't get the results from the genome annotation. But quickly, I'll show you something from another course that does use this model. So if we go to, this is for a class exercise. If you were interested in the model, you'd be looking for in your genome annotation results AMR-SIR.txt. I changed it here to a TSV file that allows me to make it into that interactive table. I make first row column headers, same thing. My antibiotics that were included within the predictive model. Prediction scores just relevant to the model itself, 0 being susceptible, 2 being resistant, 1 being intermediate. Then you have your average accuracy of your predictive model and confidence intervals for low and high. You can reorganize by accuracy and then look at the average accuracy, and look at your top resistant, your top susceptible. All right. And we are right at time. It's incredible. All right. That's everything I wanted to cover for today. Next week, we're going to be starting off using those mags that you created today to go a little bit deeper to comprehensive genome analysis. We're going to build phylogenetic trees, do some comparative genomics. But at this point in time, that's everything have here. So if you have any questions, I'll stick around. At the very end of your handout, I have hyperlinks for service tutorials if you want to know a little bit more about the services we covered, and a manuscript from our recent publication that includes a little bit more about that predictive model that we talked about at the very end. So that's all I have. Feel free. Okay, got a couple questions. Let's see.
I can read it to you if you want. The heat map
Yeah.
and what job output file from taxonomic classification did you select to view the heat map?
Yeah, so for the heat map, I hadn't showed you because for this exercise, we only have one sample. Now, if you were to upload, when I say, I think you probably know, but taxonomic classification has the ability to accept multiple selected libraries. Here we did one, say you have 4 or 5 different selected libraries. You would need to have more than one to get the heat map, which is going to be showing beta diversity between your samples. So for example, we have an exercise from a course. They look at multiple samples with the taxonomic classification service. So if I go here, if I look, I would see, because I have multiple samples, now I can compare beta diversity. I get this beta diversity stats heat map. It shows a heat map of Bray-Curtis similarity, where you can look at, samples that are similar to each other are going to have a lower value. Samples that are further different from each other, you can think of it as the distance matrix, are going to have a higher value. So another output you might be interested in if you were uploading multiple samples. So let's see. So I see Genevieve want to ask about the TEPI community is and how people can join. So the TEPI community is going to be instructors, educators at different institutions looking to implement a Tiny Earth course within the respective institution. So to get involved, there is you can look up Tiny Earth Network. Mariah Knowles at the very end mentioned about this training, TEPI training that they would be having in the summertime. So to become a TEPI, you would need to do a training event at UW-Madison, which I think is, as long as you get accepted, I think they cover everything, but don't take my word. You're going to read about it, but I think they do have housing and everything like that. I believe it's 1 to 2 weeks long, but they'll go over everything we talked about within the course. How do they collect samples? How they do culturing in the lab? How they biochemical tests? They'll pretty much run through it like you were a student within the lab, learn about the program itself, and that way you could then implement it within at your institution. So did I mention something? I saw the heat map, and then are there plans to add sea ores, fungus, and outside the scope? Okay, thank you, Rebecca. Um, no problem, no problem. Let's see, is there any tool to view alignment to reference from a BAM and BAI files? It's a good question. Do we have tools to align for BAM and BAI files? No. Let's see.
You can use the variation service, but
Mhm.
with long reads, let me think, with long reads, you should be able to get that info. I work with short reads so much, I don't even think about that. But I think you
Mhm.
can do alignment in FASTQ utilities,
Mhm.
which should get you there.
Mhm. Yes, under the drop-down menu in the
Yes, you
pipeline.
can get align under that. So that's one of the pipelines there. And you would just align it to the reference. And I know that they just recently put in an aligner, I think Minimap 2 for long reads. So it should be able to handle both short and long reads for alignment. What I like about the variation service is you can actually see them mapped, and you can see the BAM cert files too mapped to the genome.
Mhm. Right on. Very, very cool. Thank you for that input, Becca. Let's see. Any more questions?
Jamal, someone earlier asked about the recording being available. Will it be?
Yes, I will send out the recording to everybody. So I got a decent amount of emails from individuals who were able to attend this event that would like the recording. So I'll for sure send it out to everybody with the slides as well from the beginning.
Hey, Jamal, you could also post those in the workspace that you're using. It will take videos and files, and you can do that, and they can get it there as well.
Oh, okay. All right. Um, I'll do that then. I'll put the recordings there.
Because
And
sometimes they're too big and you can't email them, so
Mhm.
then they can just draw on them there.
Okay. All right. That's a great idea. I'll definitely add it to the workspace. Just in case anybody misses it, I'll include it in the email, but that's great, especially if there's any trouble for me to get that sent out. So thanks for adding that. Right. About the workshop at Argonne, when is the last day to apply? Any information on the last day to apply to the in-person workshop?
Sooner rather than later. Processing takes a few weeks, and so I would say probably in the next 2 to 3 weeks would be ideal.
And I didn't put that in the chat. I thought I saw someone drop the link in there earlier. But it is very simple to register for it too. So no problem. Have a great weekend. So yeah, I think you just like you put your name, you're gonna add a email for a representative at Argonne that will get you approved. So it's very quick for you to put in to register for. No problem. Thank you for attending, for the kind words.
Great job, Jamal.
Thank you. Appreciate it. Appreciate it. I'm gonna go ahead and stop the recording. Yes, no problem. Thank you for attending.
Yeah, that was great.
Thank you. I need some water. Talking. That was it looks to be